So today we are starting working through the paper 12, paper 1 version 2 from 2017. Specifics, it is the May-June 2017 paper. Starting from the beginning, which organelles are enclosed in a single phospholipid bilayer, a single membrane. Remember, one membrane is a bilayer, and contain hydrolytic enzymes. Well, there's the giveaway clue. Hydrolytic enzymes is your lysosomes. And they definitely have a single membrane. So we head to our answers and we select lysosomes. Answer for question 1 is C. Question 2 asks us, the DNA of prokaryotes is naked and circular. They just got that loop of DNA. Of however it's going to sit in that specific prokaryote, okay? No proteins associated. Which statement describes how the DNA of eukaryotes, so now we have our true nucleus, differs from that of prokaryotes? So, it has a nuclear envelope around it and it's a double helix. It has a nuclear envelope around it and it's circular. It has proteins attached to it and it's a double helix. It has proteins attached to it and it is linear. So this is one of those ones where you've really got to think nicely. What can we knock out immediately? Nuclear envelope and circular. We don't have a circular DNA. So remember this bit here that says differs from that of prokaryotes. Well, our prokaryote is naked and circular DNA, but it is still a double helix. So our double helix options can also be crossed out because we're not just describing eukaryotic DNA, we are describing how it is different from the prokaryote. So something that they have in common doesn't apply. So we're left with proteins attached and linear, which is correct. Our eukaryotic DNA is stabilized with that addition of proteins and it's cut into linear chromosomes. So 2 is D. The recently discovered Pandoravirus measures 1,000 nanometers in diameter. The mimivirus has a diameter of 400 nanometers. What can be detected using a light microscope with a maximum resolution of 0.25 micrometers? Now remember, for things to look at resolution, we have to be going in terms of what's going to interrupt that wavelength. So 0.25 micrometers would be 225 times 1,000. 0.25 times 1,000. 1, 2, 3. Yeah, 250 nanometers. Okay, that's our maximum resolution. So, which of these two viruses would disrupt that? Well, 400 and 1,000 are both bigger than 250. So we would be able to see both of them. Question 4. What are found in chloroplasts and mitochondria? So it's important that we're using that and. Okay, do we have DNA in both? Yes, we do. Do we have 70S ribosomes? Yes, we do. Do we have mRNA? Now that's not something that we study directly, but we need to think. Okay, chloroplasts and mitochondria have DNA and they have ribosomes. So the only way that the instructions could get from the DNA to the ribosomes in order to build the proteins that these organelles need is with mRNA. So answer is A, 1, 2, and 3. In order for chloroplasts and mitochondria to build proteins, they're going to need that DNA, 70S ribosomes, and the mRNA. Question 5. Boiling the bones and teeth from dead animals can be used to produce a type of glue. The glue is formed from the collagen fibers present in bones and teeth. Which statement describes the changes to collagen that occur when the glue is produced? Right, so we're talking about collagen fibers and we're talking about boiling. So protein structure is coming in here. So we're looking here at what are our different options. The fibers of collagen become more coiled. The fibers of collagen become more flexible. The helices of collagen molecules unwind, or the molecules of collagen dissolve in water. Okay, so we can take out the last one. We know that's not right. So when we're talking about boiling our protein, is it going to become more coiled, more flexible, or unwind? Okay, boiling is heat, is more movement, so we're not going to become more coiled. When we're becoming more flexible or unwinding? Well, you know about the breakdown of protein structure, and you know what happens is that protein falls apart. We lose the bonding. So we don't talk about flexibility in terms of proteins. What we're going to be talking about is the unwinding of those helices. We've had a question come through about mRNA and RNA. So remember, just mRNA is one type of RNA. Your RNA splits into mRNA, tRNA, and rRNA. Each of them has a slightly different structure and a slightly different role within the cell. Moving on to question 6. What describes the primary structure of a protein? Protein structure is really important. It's really important that you know the details of what is each level of structure and how do they relate and how are they formed and what bonds are involved. So primary structure of a protein, that is our amino acid sequence. So is it alpha helix, a dipeptide, a globular structure, or the specific order of amino acids? Definitely specific order of amino acids. Alpha helix would be secondary structure. Globular would be tertiary structure. Dipeptide is just 2 amino acids bonded together. So if you have questions, you can send them through either on the Q&A or through the chat. Question 7 tells us that the diagrams show 4 monosaccharides with a formula C6H12O6. This is looking familiar because this is our glucose. Which diagram shows alpha glucose? Remember, you've got alpha glucose and you've got beta glucose. And it's all about the OH on carbon 1. Alpha, we have the OH down. Beta, we have the OH up. So they want to know which one is alpha glucose. So first off, OH is down on carbon 1. So this one is out, the H is down, OH down, OH down, OH down. Okay, so which of these 3 is our alpha glucose? So we know that beta behaves, which means that we have H, OH, H, OH in a nice and reliable sequence along the bottom. That helps us remember that our alpha is going to go OH, OH, H, OH. So if we look at all of these, we can see that we have OH, H, OH on B, we don't have it on C, and we don't have it on D. So that confirms to us that B is our alpha glucose. So it's really important that you do know the structure of glucose because it comes up in many different ways. It's really important that you actually know everything. Question 8. Complete digestion of polysaccharides requires all the glycosidic bonds between the monomers to be broken. Amylase only breaks alpha-1,4 glycosidic bonds. Which row shows how completely amylase can digest molecules of cellulose, amylopectin, or amylose? So we need to think about who is branched and who is not. Because 1,4 glycosidic bonds, this makes our chain, our basic connection of those molecules. If we have branching, then we have also 1,6 bonds. Okay? Then we've got to bear in mind that it's only breaking alpha. So if our carbohydrate is made of beta glucose, then it's not going to work because enzymes are so specific in shape. So our beta glucose is a different shape. An enzyme that's specific for alpha glucose won't fit onto beta glucose molecules. So we have cellulose, we have amylopectin, and we have amylose. So we know cellulose is beta, amylopectin and amylose are alpha. Okay, so we're not going to digest cellulose at all. So C and D are out. So the question is amylopectin and amylose. Which one is some digestion and which one is most digestion? In other words, some digestion will be our branched molecule and most digestion will be our unbranched molecule. Because our unbranched will only have 1,4, whereas our branched will have 1,4 and 1,6, and those 1,6s won't be broken. So who is branched out of amylopectin and amylose? It is amylopectin. Amylose is unbranched. It's the one that coils up. So amylopectin must have some. Amylose is most. Our answer is B. And it really helps when you're working through these papers, even your final paper, to actually interact with the paper like this. Write notes. Make sure you sort of knocking out answers, focusing on the potential right answers, really working through the information in the question, and tying that in with the information in your brain. Question 9. The diagram shows the results of tests on 4 solutions containing biological molecules. Which shows the solution that only contains only starch and protein? So what is everything testing for? Burette test going purple, then this is protein present. Iodine test going blue-black, this is starch present. Benedict's test going orange, this is reducing sugar present. But they've told us that it's only starch and proteins. So we're going to take out our reducing sugars circle because we don't want them present. So D is in both circles, A is only in the starch. So the answer is D. Starch and protein are present in that solution. Question 10. The enzyme invertase catalyzes the breakdown of sucrose to glucose and fructose. 3 different enzyme inhibitors of invertase, X, Y, and Z, were investigated. The percentage inhibition of invertase was measured at different concentrations of inhibitor. So we've got the concentration of inhibitor, and we look at how much it inhibited. So increasing concentration. So we have 1, 2, 3, 4, 5 different experiments, for example, for X, for Y, and for Z. They then ask us which are valid conclusions from these results. So percentage inhibition, okay? So this means that at the top here we have high inhibition, whereas down here we have low inhibition. So it's a little bit different from the graphs we normally look at for inhibition. So we need to take the time to understand. So inhibition would mean very little product. So inhibitor X at high concentration has high inhibition. Inhibitor Y at high concentration, sort of high. Inhibitor Z flattens out. It has sort of medium inhibition, let's call it, all the way from 4 up. Okay? It doesn't get any more inhibiting. So let's look at our possible conclusions. The higher the concentration of inhibitor X, the less sucrose is broken down. Well, that's true, because we have very little product at high X. The production of glucose and fructose using inhibitor Y is higher than when inhibitor Z is used. So we have to now consider production of glucose and fructose using inhibitor Y is higher than when inhibitor Z is used. We don't have any information on the production of glucose. We only have information on inhibition. So we can't actually conclude about production. Production of glucose and fructose at an inhibitor concentration of 2 arbitrary units is lower than at an inhibitor concentration of 4 arbitrary units for all inhibitors. Okay, let's break it down. Production of glucose and fructose at an inhibitor concentration of 2 is lower than at an inhibitor concentration of 4 for all inhibitors. Well, it is suggestive of the relationship, but we've got that same problem. It's production of glucose and fructose. So we definitely confirmed that we like 1. So we can knock out C and D. So then we're down to 1 and 2 or 1 only. And the problem with 2 is that production story. So we are going to go with B, 1 only. We can also remember that production of glucose and fructose with Y is only bigger than Z from 4 to 8. See here, Y is only bigger than Z from there. At this stage here, Z is inhibiting more. So they're actually flip-flopping their relationship. At low concentrations, then Z is more effective. At higher concentrations, Y is more effective. Let's finish that. So it's really important that you break down each of these very, very firmly so that you know exactly what it is going on. Question 11. The following statements are about enzymes. Folding of an enzyme molecule causes the formation of an active site. The shape of the active site changes to enable the substrate to bind. Temporary bonds hold the substrate in the active site, and more enzyme substrate complexes are formed at the optimum temperature. Which statements are correct for the induced fit hypothesis? So if we limit ourselves to thinking about that induced fit hypothesis, folding of an enzyme molecule causes the formation of the active site. That has nothing to do with the induced fit hypothesis. That is just how we're forming that active site. The shape of the active site changes to enable the substrate to bind. That is everything. That is our induced fit hypothesis. Temporary bonds hold the substrate in the active site. Definitely. And more enzyme substrate complexes are formed at the optimum temperature. Yes, that's the whole idea of induced fit. So our answer here is C, 2, 3, and 4. Question 12. Continuing with enzymes. How is the Michaelis-Menten constant, Km, used? Is it used to assess the efficiency of an enzyme in catalyzing a reaction, to compare the affinity of enzymes for their substrate, to find the maximum velocity of an enzyme, or to find the rate at which substrate is loaded by an enzyme. Km is all about enzyme affinity. So the answer is B. We can compare affinity of enzymes for their substrate. So we can have that in terms of, if we have two different enzymes that catalyze a reaction, we can look at the different affinities between them and then select the one that we want. We've had a question coming through about hypotheses being, is the hypothesis the possible answer for the investigation? So a hypothesis is what you said, yes. You would hypothesize that this would happen or that would happen, and then you would do your investigation, your experiment, to answer that question and to determine if your hypothesis was correct or not. For question 13, we're moving away from enzymes and we're moving to cell surface membranes. Proteins in the cell surface membranes of human cells and mouse cells were labeled with fluorescent dyes. The human cells were labeled with a red dye and the mouse cells were labeled with a green dye. A human cell and a mouse cell were then fused to form a hybrid cell. So you had your human cell with proteins, you had your mouse cell with proteins. Now you've fused, so you've got human proteins and mouse proteins. What's going to happen? At first, the different dyes remain separate, like we've drawn here. After 40 minutes, the two dyes were evenly distributed in the hybrid cell surface membrane. So now we've got like this. What explains this observation? All protein molecules in the cell surface membrane are fixed to structures within the cell, but phospholipid molecules move freely between them. Protein molecules are not fixed. Groups of protein and phospholipid molecules in the cell surface membrane are attached to each other and move together. No. Everything moves freely in that cell surface membrane. Only protein molecules in the outer layer of the cell surface membrane can move freely between phospholipid molecules. No. We are not limited to the outer layer. Everything in the cell membrane moves freely. Proteins, phospholipids, both layers. Protein molecules in the outer layer of the cell surface membrane and those which span the bilayer can move freely between phospholipid molecules. So there it is. The final answer is the correct one for this question, which is a good thing because we were running out of options. And then question 14. The diagram shows a plant cell. So nucleus, vacuole, cytoplasm in gray, cell wall around the outside, double line. The plant cell is put into a solution with a water potential less negative than the cell contents. What will happen to the appearance of the cell? So we've got a few things going on here. We've got the vacuole shrinking or enlarging, and then the cell contents, cytoplasm staying the same, cytoplasm pulling away, cytoplasm pushing against the cell wall. See how it's sort of slightly rounded, and the whole cell growing. So we can knock out D. We know that that cell is not going to grow like that. So now we need to think about the solution is less negative than the cell contents. So water potential, we move from a high water potential to a low water potential. That's what we're going. So this is from pure water, and this is concentrated. So let's put our cell here, and the water potential is less negative. Now, if you're less negative, you are closer to 0. So you are here. This would be our solution. So now we can see that the water is going to move from the solution to the cell. So this loss of water from the cell that's represented in A and B is wrong. The answer is C. Water is going to move from the solution into the cell. So the vacuole will enlarge and the contents of that cell is going to press against the cell wall. Question 15. The diagram shows the fluid mosaic model of membrane structure. So we can see our phospholipids with their heads and tails. We can see cholesterol molecules. We can see a glycolipid and a glycoprotein and a protein. So they ask us which molecules in the membrane are identical. That is a very strange question. So they ask us, 1, 2, and 3, are they identical? No, that's a protein, that's a glycoprotein, that's a glycolipid. 1 and 4, we have a protein and a, sorry, it's gone out of my head, I just said it, cholesterol. 4 is a cholesterol. So we have a protein and a cholesterol. They are not identical. 2 and 3. Glycoprotein, glycolipid. Not identical. 4 only. Well, it's the only thing that makes sense. It's not identical to anything else, but it is identical to the other cholesterol molecule that is in the membrane. And that has taken us to time. We'll continue with the paper in the next lesson.